No, thanks. I'm happy to be here again. I was here last year. A word first, just after that teaser, a little bit more on the book, which posits, and I believe this, that within 20 to 40 years, most people with good health coverage who want to be parents will conceive in a lab. They will make about 100 embryos. Each embryo will be whole genome sequenced. The parents will then be asked, what information do you want about these embryos? And based on the information that they get about disease, about cosmetics, about behavior, about sex, they will choose one or two to be transferred for possible birth. Cheap whole genome sequencing is essential to this, but the key technology is stem cell technologies to be able to turn induced pluripotent stem cells into gametes, which has already been done successfully in mice, and steps toward at least primordial gametocytes have been done in humans. So I think most of that 20 to 40 years, I think, is probably regulatory safety testing rather than actual science. And the book spins out what the implications are, at least as I see them, both good and bad. It was fun writing it. I hope some of you will read it at some point, because it'd be a real drag to have spent 5 years writing it and have only Dana read it. And he hasn't even finished it. But to CRISPR, which doesn't have much to do with my book, although it did cause me some heartburn. In the original versions of the book, I said, well, I'm talking about embryo selection. You could do embryo editing, but that's going to be much farther off because the technology is far away. And then over the last year and a half, as I became aware of CRISPR, I had to rewrite and rewrite and rewrite. I still think it is farther away than embryo selection. Embryo selection's been with us since 1990 in the form of pre-implantation genetic diagnosis. But these folks made me rewrite. They also got me involved and interested in this area of genome sequencing. One of the many meetings that I've been fortunate enough to go to was one organized by Jonathan and Dana and Jennifer and others in Napa Valley in January of 2015. So only a year and a half ago. 14 of us got together around a room to talk about the issues arising from CRISPR, particularly but not solely in the context of humans, which led to an article in Science, which led indirectly to the great CRISPR Human Genome Summit of December 2015, and has led to lots of continuing controversy, and has brought me more in touch and gotten me more involved and interested in what I think are the genuinely transformative possibilities of genome editing. I want to talk about those in 3 categories. And in each category, there's a bit of a dualism. So the 3 categories are: first, human germline genome editing; second, human non-germline genome editing; and third, non-human genome editing, collapsing the germline and the non-germline in that case. The additional bifurcation that's overlaid on all this is medical versus enhancement. In each of those 3 categories, the difference between medical and enhancement plays some interesting roles in the ethical, legal, social implications. I'm a law professor by training. I'm a lawyer by training. My last bio class was in 10th grade when we barely knew how to spell DNA. I remember dissecting the shark. That was fun, but that's kind of all I remember from 10th grade biology. And I don't think of myself as an ethicist, but what I'm interested in are the social effects of biotechnologies. The effects on the law, the effects on society, on how we live and organize ourselves, how we think of ourselves, and of things that people think about as ethical questions. I tend not to come at it with a strong normative view of my own about what's ethical and what's not, but I have a pretty good sense of what my society thinks is ethical and isn't. And when scientists and others do things that their society thinks are unethical, bad things tend to happen. So with that framework, let me talk first about human germline genome editing. And I should say I now have 2 ex-teenagers. Having ex-teenagers is a lot better than having teenagers. So I'm perfectly used to being interrupted at any and all times, and to being denounced as an idiot, etc. I still remember it. It's now a little bit in the past. I've gotten a lot smarter since my kids are in their 20s. It's really quite amazing. But if you guys have any questions, feel free to ask. I will try to keep some time at the end for questions, and then I think Jonathan and I are going to try to have a broader discussion in the next section. So, human germline genome editing. Most people think about this as human embryo editing. It's really broader than that. I think that's a mistake. In fact, I think if we ever do it, we're unlikely to do it on embryos. For one thing, with the embryo, you've got problems of mosaicism. You've got to make sure that all of the cells of the embryo, or at least all of the relevant cells of the embryo, get changed. I think if we do it, it's likely to be to gametes, change the eggs, change the sperm, whether in vitro or in vivo, in somebody who's alive. That way you can check to make sure the changes were made properly, that they weren't off target. If you've got a nice cell line of spermatogonia or primordial eggs going, you can see whether the changes have been made in the right places. You don't have any off target stuff, and then make your gene edited baby, embryo and baby that way. But whether it's through gametes or through embryos, people are really exercised about the idea of generation passing of transgenerational genetic editing. Most of our meeting in Napa in January of 2015 was devoted to discussing this. That meeting was really impressive in that, for lots of reasons, but one was two of the people at the meeting were Paul Berg and David Baltimore, both Nobel Prize winners, but perhaps more relevantly for this, two of the 5 organizers of the Asilomar Conference on Recombinant DNA, which had actually been held 40 years to the week before our meeting in Napa. And they had kind of a strong reaction to it. They said, we promised we wouldn't do this. When we did recombinant DNA, we promised we'd never mess around with the human germline. And that was an easy promise to give because we didn't have any capability of doing it and didn't think we were going to have the capability of doing it. And having we, it wasn't quite clear to me whether we was Paul and David, or we was science as a whole, or a particular generation of scientists. But they took that very seriously. Lots of other people have taken the idea of changes that go down through the generations very seriously. That is what led to the December 2015 summit that the National Academies of Sciences called and held in DC, along with the National Academy of Sciences of China and the Royal Society of the UK, that several of us were privileged enough to be at. Almost all of the discussion there was about human germline modification. People are worried about the permanence of germline modification. People are worried about the arrogance or the hubris of germline modification. Do we really know enough to control our own genes, etc.? People are worried to some extent about consent issues. The babies that are born this way didn't agree to it. This is probably the hottest button issue about CRISPR in the country and the world. A number of countries have already passed laws banning it. Arguably the European Council, which governs it's not exactly the EU, but which has power over a whole variety of European countries, has passed a convention banning human germline modification. So people take it very seriously. I don't. I don't think it's very important for a variety of reasons which I hope to lay out. First and most important reason is we care a lot about safety when we deal with people. If you are to make babies in a brand new way, it better be safe. Even in countries that don't ban it, like the United States, the FDA is going to take the position that that modified embryo or the modified gametes are more than minimally manipulated cells and are hence either biological products or drugs or possibly devices. But whatever they are, you can't use them without proving to the FDA that they're safe and effective. Can't use them generally, can't even use them experimentally until you've proven to the FDA that they're safe and effective enough to warrant an investigative new drug exemption. And although there is the statutes from 1938 that govern the FDA, the Federal Food, Drug, and Cosmetics Act, doesn't talk about making gametes or editing genomes, editing embryos. The positions they have staked out that have been upheld so far in court easily can be applied to this sort of world. And if they aren't, if a court were to say, no, FDA, you don't have any jurisdiction about it, I guarantee you Congress would either ban it entirely or give FDA jurisdiction within 24 hours. So it's going to be regulated for safety. It's also the case that you need people who've got skin in the game to make humans. You need a mother or a prospective mother who's willing to take part in this experiment. You need an IVF clinic that's willing to take part. You need a physician who's willing to be the obstetrician involved. You need scientists who are willing to try this. You need to worry, if you're an institution, about the possible bad public relations and the possible lawsuits. And if anything is wrong with that baby, you need to worry a lot about the lawsuits that are going to follow, as well as the public relations hit. We are currently within 2 weeks of the 38th birthday of Louise Brown, the world's first test tube baby, born in late July 1978. There are now 5 million in vitro fertilization babies, some of them 37 years old, alive around the world. If Louise Brown had not been a healthy baby, that technology would have taken a lot longer to get going. So those are concerns that exist with people that don't really exist with non-humans. If you have a system where 99 out of 100 of the little mosquito babies, mosquito pupa, don't survive to adulthood, no one is going to be upset. Most people will cheer. PETA does not care about mosquitoes. So we do care about humans. So the constraints, both the legal and regulatory constraints on the one hand, and the more social, public relations, people who need to be involved with their own, who need to put their own reputations and professional futures on the line with humans are a lot greater, which makes me think this is going to go very slowly.
But are fertility clinics regulated?
Not very much, but but the FDA has said that fertility clinics can be regulated. So the FDA has taken the position that mitochondrial transfer is regulated. They've taken the position that human cloning for reproductive purposes would be regulated. The one thing they do right now, the fertility clinics do, that the FDA probably should, under its theory, regulate, but doesn't, is something called ICSI, intracytoplasmic sperm injection, where they grab a little sperm, they break its tail. I've seen videos of this. It's really hard not to anthropomorphize it when you look at the video. The poor little sperm has its tail broken, and then the poor big egg is sort of forcibly violated by this syringe going through it. It's very odd for me to see myself anthropomorphizing these two cells. But that's probably something that the FDA should have taken regulatory jurisdiction over, but it happened in the 80s when the FDA wasn't thinking about it. You want your tail broken off? So I think it's not all that likely to happen quickly because of the government, as well as the regulatory and government pressures. Secondly, though, and this is on the medical side, there's almost no good reason to do it. Almost everything that you could do medically by genetically editing an embryo, you can do through a well-established, safe and effective method called pre-implantation genetic diagnosis. The heart of my book is also part of this issue. If you've got two parents who are carriers who are heterozygous for an allele of the CFTR gene that causes cystic fibrosis, they have a 1 in 4 chance that any of their embryos and children will get CF. It's actually a little less than that because CF turns out not to be 100% penetrant, even though we thought it was before we could genotype and sequence people. So you could edit their cells to get rid of the CF alleles, or you could make embryos, test them, and say, okay, these embryos, haven't inherited 2 bad copies. They're safe. These embryos have 2 pathogenic copies. We will not transfer them. That's been done for over 25 years. To the extent you're talking about autosomal recessives, most of those go away. To the extent you're talking about autosomal dominance, again, somebody with Huntington's disease wants to have children who won't have Huntington's disease. Each of his eggs, his sperm or her eggs, has a 50-50 chance of getting the Huntington's allele. One allele causes the disease in the offspring. You don't need to edit their eggs or sperm, let alone the embryo. You just need to make some embryos and find the ones that don't have the bad copy and transfer those. And it should be, on average, 50%. Now, some have said, well, you know, what if you only get a couple of eggs? And by bad luck, they happen to be the ones that have the Huntington's allele. Well, my response to that is try again. Get some more eggs and do it again. And 50-50, you can roll, you can have the bad roll of the dice 10 times in a row. That'll happen once in 1,000. Okay, in that case, do it 11. Do it 12. There's no reason in that circumstance to go forward with gene editing. Gene embryo selection will do the job. Now, for the sex-linked disorders, which are mainly X chromosome linked, same story applies. They can only come through, they typically come through the mother. And the mother has 2 copies. You can figure out which one's getting passed on. The father also has an X. If the mother's homozygous on her X for the problem and you want a boy, then you might need to do editing because the mother can only pass on the bad copy and the father isn't going to pass on, with a boy, the father's going to pass on the Y and not the X. So there's one exception. How many of those are out there? I don't know. Here are the exceptions that I know of. If you have an autosomal dominant disorder and you are not heterozygous but homozygous for it, and there are people out there who have 2 bad copies of the Huntington's allele, they tend to be early onset, but they make it, many of them, into their teens and 20s. If they're well enough, they live long enough and they're well enough to want to have kids, the only way they can have a child without Huntington's disease is genetically related, a child who has half of whose genome comes from that person, is to gene edit. Because they got 2 bad copies, they're going to pass one on, it only takes one to make the disease. How many such people are there in the world? More than 0, but probably not very many more than 0. So you're talking about a serious disease. 1 allele makes you sick, 2 alleles, you've got to live long enough and be healthy enough to want to be a parent. It's not going to be very many people. The second exception, I think, is a little bit larger. That's situations where 2 people want to have kids together. They both have the same autosomal recessive disease. So imagine 2 kids with cystic fibrosis who meet at CF summer camp, fall in love, want to have children. All they've got to give are mutated versions, are pathogenic versions of the CFTR allele, or the CFTR gene. So all their kids will get that. And if they want to have children who won't have CF, they need to do gene editing or use donor egg or donor sperm. But if they want genetic children, quote, children of their own, a phrase I really don't like because it denigrates the role of adopted parents and parents who've used sperm donation or egg donation, but it's the common term. If they want to have kids of their own without CF, they're going to need to have gene editing. Third category is mitochondrial disease. I leave mitochondrial disease out a little bit. Mitochondria only pass down through the woman. I think mitochondrial transfer is very likely to be a much more quickly available, safe, and effective remedy for that than trying to edit the mitochondrial genome. So 2 main issues, 2 main sets of people who need this for medical purposes. People with an autosomal dominant disease who are homozygous for the bad allele, and 2 people with the same autosomal recessive disorder. How many people in the world is that? How many couples in the world is that? I'm willing to bet it's more than 100, maybe more than 1,000, more than 10,000? Kind of doubt it. 7.3 billion people, not very many who need to do this. And to preview a little bit, that still implies that in order to avoid having a sick kid, you would need to change the germline genome. If somatic cell gene therapy using CRISPR or other techniques gets good enough, you can have the child born with 2 CFTR mutant alleles, pathogenic alleles, and fix it later. So even then, you may not need that in order to have a healthy child. So that's the reason I don't think there should be a big demand for this medically. I'll come back to the enhancement side. But medically, there aren't that many people who are really going to need it. George Church makes an argument that gets more and more attenuated and more strange, that there are other categories, and he's right about some of them, like an X chromosome linked one where you want to have a boy, but I don't think it's going to be a big deal. So there's not as strong a driver, and the health of the child is a big driver for parents to be willing to do this, as well as for doctors and IVF clinics to be willing to participate, as well as for governments being willing to allow it. And that doesn't disappear entirely, but it pretty much goes, it's quite minimal.
What if I believe an embryo is a citizen? Don't I have a right to genetic health care for all of my children?
You have a right to believe anything you want. In the United States, embryos are not citizens, fetuses are not persons, and even living persons don't have a right to health care in the United States. Now, in civilized countries, you do, but not here. We're getting closer, but we're not there yet. But yes, if you believe that, then you can make that argument. Good luck getting a government to buy it. Maybe you can find some scientists in IVF clinics that will buy it. Yeah.
With the advances in genomics as we go through time, with like better sequencing, better mutational algorithms, whatever, we might learn a lot more about genetics. You're saying 40 years ago, Bird said, well, we don't have an ability to do human genome editing, so we could say, we're never going to do that.
Right.
Now we do have the ability to do that. What about once we have the ability to understand more complex diseases that are genetic, but they're not autosomal, and it may not be as simple as just picking an embryo, depending, I'm not sure. But I'm saying that what you're saying is that based off our current state of the art, what we know today, and the things we can actually address, which are probably autosomal type Mendelian diseases, there's not going to be a demand for this. But do you not expect there to be a demand for this once we understand this?
So I think that's more relevant on the enhancement side than on the medical side. To the extent you've got multigenic diseases, and obviously there are a lot of disease risks that are multigenic, that also means it's a lot more disease alleles to pick and choose from among the embryos. Sure. So if it takes 10 genes to give you a disease, that's 10 that all have to line up the right way, which means it should be even easier to find unaffecteds. It could be that the world, empirically turns out to be different. It could be that there are a lot more autosomal, powerful, highly penetrant diseases than we expect. Hard to believe at this stage in our knowledge of genetics. And it could be that some of the multigenics would be susceptible to gene editing, but not susceptible to embryo selection. But I think that's likely. I don't think that's very likely. But a lot of this is open empirical questions. Yeah.
About ApoE4, where roughly 1 in 4 couples will have one of the two be homozygous. So if you're homozygous, you have a 30-fold chance it's almost certainty chance of getting dementia at 75. But even if you get one,
if you live to 75, but
you get a 5-fold, but most people will. But even with one of the two alleles, 5-fold increase.
5-fold is high from the estimates I've seen, but it's certainly significant. I've seen double or triple average risk, less than 50% still. So that's an interesting example. ApoE4, you can think of it as a incompletely penetrant autosomal dominant disorder that's almost completely penetrant when it's autosomal recessive. So I can put it into those two categories. It's only 2 couples, both of whom are homozygous for the ApoE4, who would need it to avoid having a homozygote.
Which isn't so rare at all.
It would be about 4 out of 10,000.
Is that right?
Yeah, it's like 2% of the population, so 2% squared. Yeah. So it's not terribly rare. We don't know whether there's any die-off or fertility or other issues earlier on, but I think it's 4 out of 10,000. But I'm a lawyer, so don't trust me on numbers.
Jonathan's going to look it up.
Yeah, I know he is. He's pulled out his
But
phone.
it's much, much bigger than all these other ones that we're talking about.
Right. And then you get into the question of disease at age 75, how do we value it, particularly when you've got 75 years of future research to look for preventions and cures. 75 years is a long time. I'm not expecting an Alzheimer cure in time for me, but in 75 years, I'd be really surprised if we don't have ways to prevent it. So yes, and there will be other examples like that, but I don't think they're going to be that many and that powerful.
I guess at the heart of it is this different than safe and effective, or a positive risk-benefit analysis?
So, and I want to come to that, but that's not the part of the argument I'm making right now. The only part of the argument I'm making right now is to say there shouldn't be strong medical demand for this, because the medical genetics, reproductive things can almost all, not all, but almost all be taken care of through embryo selection, which is a much more straightforward, well-proven, well-established procedure, rather than need to go to gene editing. And this is an argument not about ethics, it's an argument about markets and drivers and things that are going to push people towards something. You will find early adopters who will push through when there isn't a strong need for it. But for something to become well established, I think there has to be a strong need. People don't do IVF for the most part just because they want to do IVF. They do IVF because it's the only way they can have babies, or the only way they can do PGD to have babies who won't get Huntington's disease when they grow up, et cetera. So you need a strong driver, and I don't think there's a strong driver here. But on the other hand, I think the talismanic nature of the germline is empty, is almost bizarre. People care so much about the germline, and they talk, some of them, about the idea about the human germline genome. Well, let's think about that for a second. Whose human germline genome is the human germline genome? Obviously, it's mine. You guys may disagree. There's 7.3 billion human germline genomes. Maybe a little less for identical twins, but that's rounding error. If you're thinking about haploid genomes rather than diploid genomes, there are 14.6 billion of them. Even the identical twins are going to have some differences post-conception in their genomes. So each of those is a separate human genome. The human genome isn't a thing, it's a cloud. And even if you're only thinking about the open reading frames, the 23,000 protein coding genes, it's an open cloud in 23,000 different dimensions. It's a huge amorphous mass. Yes, it doesn't overlap in all of its dimensions with any chimpanzee, let alone any carrot or any cattle, although in some dimensions and many dimensions it actually will not only overlap but be identical with to some extent. But it's not a thing, it's not a religious talisman, it's not the essence of our species, it's a cloud. There is no such thing as the human germline genome. Furthermore, it's not like we don't change it. Every generation changes the germline genome. I have different alleles than my parents had because their eggs and sperm progenitors mutated. Every cell division on average causes at least 10 base pair changes. If they're in the eggs and sperm, and those eggs and sperm are lucky enough to make a new person, that new person has changes. It's different from its parents. Our germline genome changes generation to generation, and those changes are inheritable. And also, by the way, people quickly say, well, if we change it in this person's genome, it will go on through all the species, for the future. Well, only if that person has kids, and only if the kids inherit the edited allele. So it's not that big a deal in that sense, but our germline genome changes. It's not this static thing. The argument, as it does so often with bioscience, comes down to, is there a significant difference between changes that happen, quote, naturally, which typically means without human intervention, or at least without human intentional and knowledgeable intervention, because we can do things that mutate our germline more quickly, like radiation exposure and so on. So we can intervene, and aspects of our lives no doubt intervene. I've flown 115,000 miles already this year, which if I hadn't had the vasectomy would probably not have done, would have had some implications for the genomes of any future progeny. We change it, but we don't change it intentionally. And so we see this argument a lot, and it's always puzzled me a little bit, that it's worse for us to intentionally, knowingly, and intelligently make changes than it is for those changes to happen randomly. Some of that is because the non-random ones we don't have to take responsibility for. It's nobody's fault. Nobody did it. But random change being better than guided change, even though guided change is not going to be perfect, it's probably going to be better than random most of the time. So I am not a big believer in this idea that somehow the germline is special. I think, well, with one exception, the germline is sort of special. If we do somatic cell gene therapy on me, and I think I'm competent, I know I'm conscious, I make a decision that this is what I want to try, it doesn't work out, I die, I would be sad if I were still alive to feel sad, but I would hope there would be some people who would be sad, but I've had 64 pretty good years, and I made the decision, I volunteered for it, so what? You make a baby this way, that baby didn't volunteer. So I do think the lack of consent of the affected individual, the baby involved, raises the stakes a little bit, mainly in terms of how confident you have to be about safety, I think. However, how many of you in this room remember signing the consent form to be born at all? I didn't volunteer. I'm happy with how it turned out. If I'd been assigned, if I'd ended up with different parents, I might have been less happy. Others, I might have been more happy, I don't know. None of us volunteers to be here. We're all the product of random, which sperm hits which egg when, as well as what mutations happen later, as well as all the environments we're exposed to, prenatally and postnatally. How much different is it when there is some intentional choice involved, some control involved in doing the editing? And I think a little bit, compared to the competent adults saying, try this on me, doing it on a baby is a little more serious and should require more confidence and safety, but not a lot. No, but a baby is. But a baby is, yeah.
We also didn't consent to things our parents did during their lifetime.
Of course.
Increase their genetic risk, or born late and to older parents, or I mean, I think it's
So how many of you in this room? Yeah, how many of you in this room are parents? Yeah. So how many of you have coerced your children? All your hands should be up. Not only is it inevitable, but it's our duty. Otherwise, they turn into little barbarians. They may still turn into little barbarians, but if we don't teach them our culture's ethos, morality, etc., where are they going to get it? We coerce them all the time, and the kids don't consent to that. So I don't think it's a false argument, though. I think it is not as strong as some people think. I do think that you should be able to do riskier things on a competent, consenting 64-year-old than you can on an unborn baby because of the consent. It doesn't mean you can't do them in ways that will affect the baby, but I do think you need to worry more about the safety than when there's been a consenting, competent person saying, yes, I know the risks, go ahead and try it. Now, the other step you can take is to say the parents were the consenting, competent adults, as we often are on all sorts of things we do with our kids. And that has some power. But again, I just ramp up the safety when you're dealing with the birth of an unconsenting kid.
I don't actually know the answer, but what is the legal consequences of a parent when they're pregnant, for example, drinking alcohol, doing drugs, things like that? Is that illegal for a parent to do that or not?
Certainly, drinking alcohol isn't. Some parents have been prosecuted for things like cocaine use. There have been some efforts. It varies some from country to country. In general, we let pregnant women do risky things, although The social opprobrium that can come with them in some subcultures is extreme. With our second child, my wife was 2 weeks, actually she wasn't 2 weeks, she was 2 days late, but she's a physician, so she had inside help and was able to get an induction. She was so tired of being pregnant. She was huge. The day before she's to be induced, we go out to our favorite Italian restaurant. She's 1 day from giving birth. She orders 1 glass of white wine, and the stares she got, the dirty looks she got from people in that restaurant were really quite astonishing. Even though the chance that 1 glass of white wine at that stage in development would have any negative effect was asymptotic to, I'm willing to say, equal to 0. So there aren't legal, for the most part, there aren't legal barriers, but there are cultural barriers.
Those things can have a biological effect on your child. You editing the genome, I can argue.
Sure.
Similar.
Except,
your intentions are good in
yeah,
that case.
yeah. Except we already know something about those risks, although we tend to think we know more than we do. So the crack baby epidemic turned out not to be nearly as devastating as people expected it to be. We know something about those risks. We don't yet know enough about these risks. Let me be clear. I am not arguing that this is a good reason to never do it. I'm arguing it's a good reason to be damn confident about safety before you do it. Because the consequences, and again, I think the parents in this room may feel this more than the non-parents, consequences of bringing a life into this world are serious. That's a lot of responsibility that will be with each of us, and we hope until we die, because the alternative is they die first, which you certainly don't want. We got to be really careful there. Yeah.
I think you might be underestimating the market, what you're saying. The shift in genomics, what we're seeing with systems technology, you have a lot of new companies that are doing personalized medicine, where they're tracking your genome, they're tracking what you eat, what you do every day.
Right. We call that the narcissome, or the Snyderome, if you're at Stanford.
You can call them narcissism what you want. The point is that they're going to track what they want, and the ones that are doing it have the money to do it, and they're going to want a market to be more preventative, to treat the cause, and they will treat the symptom down the line. I think it's coming a lot faster than you might be anticipating.
Maybe. This requires there to be complex, it requires there to be genetic solutions for prevention. It requires them to be ones that cannot be obtained by embryo selection from the two people involved. And that's possible. One of the examples George Church likes is CCR5, which is a molecule on T cells that's essential for HIV to infect the T cells. If you've got 2 null variants of CCR5, your T cells won't be infected by HIV. And he says, well, this is a great kind of genetic vaccine against HIV. Not clear. And also what the side effects, the other effects of missing that aren't clear, particularly since the molecule is heavily conserved throughout most, if not all, mammals, and is in 99% of people. It seems worrisome, likely that there would be something there. So it's possible you're right. It would depend on there being some nice clear-cut preventive measures that were susceptible to gene editing, that were not susceptible to embryo selection. I would argue, and yes, there are rich people who will do all sorts of dumb things. I live in Silicon Valley. I understand this. And they will try this. I am strongly in favor of an FDA regulatory regime that says, we're not actually going to let you try it unless there's some decent evidence of safety and efficacy. There are people who take the view that that's way too paternalistic. That's an argument you can take. I think it's not likely to be a winner in our society. But first you got to identify those protective things. And vaccination actually, is this nice thing that sits between medicine and enhancement. When you get vaccinated against measles, it doesn't improve your health. It enhances your immune system. So it's sort of a halfway step between medicine and enhancement, and a nice segue when I finally get to the enhancement part. Yeah?
So if the embryo is not a person, or doesn't have civil liberties, then if parents are asking, first of all, they're at a fertility clinic. They might want to be stewards of their own genomes, because if it's not a person, then it's actually their genomes. And maybe they want to have those genomes edited so they didn't have to come to the fertility clinic in the first place. So increase fertility.
You want to make some embryos, you want to freeze them, you want to turn them into jewelry. I got no problem with it. Well, I got a little problem with that, but I don't have any big problem with that. You want to turn them into babies? The embryos aren't persons, the babies are persons. You do things, you leave your baby in a hot car with the windows rolled up, bad things happen, you go to jail. I think that parents have responsibilities not so much to their embryos, not at all to their embryos in the US currently, to some extent after a certain stage of pregnancy to their fetuses, varying from state to state, but definitely to their babies. So yes, you're modifying the embryo or you're modifying the gametes that lead to a modified embryo, but if the planned end result of that is a baby, then you got persons. Okay, so where was I? Human genome editing isn't likely to happen. Germline editing isn't likely to happen very soon. There are no strong reasons to do it. There are also no deep, strong reasons not to do it, other than safety and efficacy. Prove that it's safe and effective. And I've got no problem with germline editing for medical purposes. I don't think people are going to need to do it very often. But on the ApoE example, that would be more people certainly than the Huntington's homozygotes. Prove that it's safe and effective. I don't have any problem with it. And I think the people who do are fetishizing this idea of the human germline genome as this permanent thing going through the centuries defining our entire species, which is just wrong. So, it's a typically wonderfully lawyer seven-handed answer. There's no good reason to do it. There's no good reason not to do it. Safety and efficacy. And what that leads me to is a sense of enormous frustration that almost all of the conversation has been about that. And it hasn't been as focused as I would like about that, but it's been on something that's not likely to happen very quickly. Now, one way the germline could expand, germline uses could expand, is through enhancement. And I take your question about the preventive issues as along the lines of enhancement. And enhancement, I think, is going to be an issue, but it'll be a bigger issue in the future. Because right now, frankly, we don't know squat about how to enhance people genetically. We know that we can predict 90% of the variation in a child's adult height by knowing the adult heights of the child's parents. We found over 100 genes that affect, or at least the GWAS says, affects adult height. If you add up, even if an additive model were correct, if you add up all the changes, it accounts for about 15% of the variation in height. It's there, there's something genetic there. We don't know what it is. We know a lot of genes that are that are involved in intelligence. The way they're involved in intelligence is if they're broken, they make you really stupid. We don't know squat about genes that make you smart. The guy who is the most out there person in this field, a guy named Robert Plomin in England, studied a bunch of hypergenius 12-year-olds, compared them to normal people, found a genetic variation that was enriched in the hypergeniuses. Nobody else has been able to replicate this, but he found it, and assuming that he's right, it accounted for, I believe, 0.4% of their variation between their IQs and normal IQs. So if we figure out how to make people taller, prettier, smarter, more athletic, etc., then the enhancement uses might be a big issue. But I think the issue there is not germline, it's enhancement. Enhancement's a question with drugs in the Olympics. Enhancement's a question with, in my day, it was using calculators in math class. All of you guys with these supercomputers that we call phones. Those are incredible enhancements, and people have issues about when you can use the enhancement and when you can't. Enhancement is a separate issue from germline genome. It applies to germline, it applies to somatic, it applies to drugs, it applies to devices, it applies to everything. And our society is deeply conflicted about issues of enhancement. So yes, it has a place in germline genome discussions, but it's not really a germline genome discussion. It's an enhancement discussion. Somatics, yeah.
For the enhancement, germline editing,
Uh-huh.
or even maybe preventative, how do you think it would affect the children that, got these sort of genetic edits compared to other children, growing up? And
Yeah.
do you think it'd be something you would tell the child, or is it something that?
I don't know. I mean, do you tell your kids that you've given them vaccines and what the vaccines do, and that the vaccines protect them from measles, which kill 1 in 1,000 kids, but 10% of the kids at their elementary school have parents who didn't care enough about their kids to vaccinate them? I think those are interesting questions, what you tell the kids. I think one of the problems with enhancement, I love science fiction. I read enormous amounts of science fiction as a kid. And I think science fiction is very productive in raising issues. I also think it's very seductive in raising false issues. And when we talk about enhancement, people think about superhumans. They think about X-Men. They think about, maybe in your lifetimes, of the youngest of you, that will be plausible. I kind of doubt it, but maybe. But not anytime soon. We're not talking about, we don't know how to enhance anybody to super levels. So whether it's through embryo selection, as I talk about in the end of sex, or through embryo editing, I think the kids that resulted from that might be 10% more healthy. They'd look more like the way the parents thought they wanted them to look. They'd be the sex the parents wanted them to have. But there's not going to be such a huge difference that we'll be suddenly in the middle of a speciation event. 100 years from now, maybe. But that's past my life expectancy, so I'm not going to worry about it. Yeah.
So from the first part, I was thinking about this example. I don't know if you count it as enhancement or as a medical treatment.
Yeah.
Color blindness is quite often, and you easily get a woman who is homozygous of color blindness. And if they want a child who is not color blindness,
Yeah.
then either you say it's medical treatment, then it's quite a big market.
Actually, if they want a boy who's not colorblind. To get a girl who's not colorblind is a lot easier. And I'm differently colored. So we talk about color blindness, like everything, there are lots of different variations of it. I'm what's called a deuteroanomaly. We have 3 different green genes, they're all on the X chromosome. About 6 to 7% of American males see green somewhat differently. I can tell stoplights without any problem. The little lights on hotel things I have trouble with. I discovered this when my wife one day said, "Why are you wearing green socks with those gray pants?" And I said, "They're gray socks." She said, "No, they're green." I asked my secretary, I was a lawyer at that point. She said, "Oh, they're definitely green." Next time I went to the optometrist, I said, "My wife thinks I'm colorblind." So they showed me all the dot charts. First 28, no problem. Get to number 29. I said, "This is the control one, right?" He said, "You don't see that 8?" So colorblindness is an interesting example, and it is, I think, on the edge between, there's serious colorblindness where you're completely red-green colorblind, or blue colorblind, which is much rarer. And that is one where, it's one of the sex-linked ones where a homozygous woman on the X would need editing to have a son who didn't have that trait.
So there is a market, but there is a need.
There is a potential market. How many women fall into that category of being homozygous is not going to be huge, but it's not going to be trivial. It's going to be 7% squared. So yeah, deafness gets into a whole set of different interesting issues, or achondroplasia. So, and I think those are actually more relevant to the selection story than to the editing story. But if you had 2 people who were deaf, who wanted to have a deaf kid, and their deafness wasn't genetic, and they didn't have alleles for deafness, and they wanted their kid genetically edited to be deaf, that I think is going to raise some complicated questions. Even though they might view that as an enhancement, the rest of the world wouldn't. So how many people are going to edit their kids for colorblindness? Especially if an alternative is to do it at the somatic cell level after the child is born. Hard to know. Depends on how safe it is, how expensive it is, how easy, safe, and expensive the alternatives post-genetic, post-birth variations are. But it could be bigger.
As you are saying, is as the demand is so low, we shouldn't worry about it. It's just not going to be a question. But if there is a demand, and then we have to face the question if we allow it or not. That comes to the law issue.
Right.
And if we allow it, then, well, that's a basic thing, right? Yes or no.
And my answer, as I stated, is if it's proven safe and effective, I have no trouble with allowing it, certainly for medical problems. When you get into enhancement, that's a different set of questions around enhancement that society has to cope with for all sorts of modalities, genetic editing being only one of them. Yeah.
What law allows versus what insurance companies allow are completely different things.
That's true. And that's actually one of the best arguments, one of the biggest arguments about trying to keep a difference between treatment and enhancement is what insurance or Medicare or Medicaid should pay for versus what they shouldn't pay for. There, the treatment enhancement distinction is really fuzzy. And it's not clear it's worthwhile in many contexts. In the payment context, maybe it is. But of course, part of that depends on how expensive the procedures are and how much you need the insurance. Yeah. So I want to talk about somatic briefly. I don't think gene editing has any new issues with respect to somatic cell treatment. It's a gene therapy. It's a method of gene therapy. Gene therapy has issues. The issues are mainly safety and efficacy. People have been trying it for 35 years. They are now finally, I think, on the edge of success, at least for some things. I expect a gene therapy to be approved by FDA either this year or next year. Doing it by gene editing rather than by other methods, arguably it just makes it a little more targeted, safer, and better. It should improve the speed with which you get to safety and efficacy. I don't think it raises any more moral questions. The question that it does raise, again, is the enhancement question, but that's an enhancement question. And whether you're doing it by CRISPR or by other forms of gene therapy or by drug, whether you're changing somebody's erythropoietin gene, changing it in order to overproduce it, changing the promoter to the gene, giving them EPO artificially, giving them their own red blood cells that have been taken out before, having them sleep under a low oxygen tent, having them train at high altitude, it all has the same effect and needs to be viewed the same way.
But you can't disqualify them if it's in their germline.
Sure you can. Yes. Well, we haven't so far. The best example of that is a Finnish cross-country skier from the 50s who had a very high production of EPO and had a lot, and of course they didn't have any idea what it was about then, but people are disqualified currently for sometimes it's no longer strictly chromosomally based, but sometimes largely genetically caused intersex conditions. That can get you disqualified, even though you had no choice about being born intersex. So we haven't...
You can't justly be disqualified. Those were unjust, and so were discriminated on the basis of someone's germline.
I tend to agree with you, but I think there's a longer argument there. So on the somatic cell thing, I don't think there are any issues, really. Other than enhancement issues, and of course safety and efficacy issues, and a whole bunch of ethics issues that I haven't talked about that I need to note that are true on all research with humans. Things like consent, things like justice. Is it only available to the rich or is it available to everybody? These are deep, important issues that exist with all biomedical research with humans. They exist here, but they don't exist here in any particularly different way. So now I want to talk finally about what I think is really important, which is the work with non-humans. Why do I think this is important? Really important? Because it's a lot more likely to happen a lot faster and a lot less regulated way. We don't care about E. coli or mosquitoes the way we care about people. We don't regulate it very much. Tort lawyers aren't looking for suits. You don't have to have volunteer mothers. We don't ask Drosophila to volunteer to provide fertilized eggs. You've got a whole set of constraints that don't exist when you deal with non-humans. There are some legal constraints, although even those tend to be limited to vertebrates. And even when they go beyond vertebrates to animals and not to plants, et cetera. I think we risk changing the biosphere in huge ways very quickly before we even know what's happening. And what makes this especially troublesome, at least in sexually reproducing organisms, is gene drive. you guys talked about gene drive in the course of this? The two-sentence, probably not technically correct version of gene drive, which is this cool invention out of back east. It's CRISPR that keeps going through the generations. So if you CRISPR a mosquito so it's homozygous for a mutant allele, and it mates with the wild type, all the offspring should be heterozygous, one mutant, one wild type. That either helps or hurts depending on whether the trait's dominant or recessive, but the next generation down will be 1-2-1. A quarter of them will be homozygous mutant, a quarter will be homozygous wild type, half will be heterozygous. What gene drive does is transform the wild type in the progeny into the mutant version. So that even though you've got mutant-mutant mating with wild type, the outcome is not heterozygous, the outcome is homozygous mutant, which can lead to something spreading through a population very fast, unless the mutant causes a real survival problem. But if it doesn't cause a survival problem, that should spread really quickly. You could change a big population very fast. How fast depends on how big the population is, how widely distributed it is, what its gestation period is. Elephants take 22 months to have babies. Changing the world's elephants would take a long time. E. coli takes what, 20 minutes to, under good conditions to divide? Changing E. coli, bad example. Drosophila, I don't know what the gestation, I don't know how long it is to maturity in Drosophila. 10 days?
For mosquitoes, which is the one people
Okay,
care most about.
yeah. So you can change a lot of mosquitoes really fast. And here's where CRISPR, I think, and when I say CRISPR, I mean genome editing. I think CRISPR is an amazing development. I've talked about it as the Model T. Before the Model T, there were cars, but they were really expensive. They broke down a lot. You needed to have your own chauffeur mechanic all the time to drive them. And there were very, very few of them as a result. After the Model T, everybody could have a car. We don't drive Model Ts anymore. But our world, especially in California, is permanently changed by the progeny of the Model T. 20 years, I don't know whether we'll be using CRISPR. I doubt seriously we will be using CRISPR-Cas9. We may use something that's descended from it. We may use a different method. But we will be using a remarkably safe, effective, cheap form of genome editing for which CRISPR-Cas9 was the inflection point. So whether it's CRISPR or something else, where it is so special is it's taken genome editing from something that would take a serious lab with 3 postdocs working for 4 months with several $100,000 to make a change, to something that 2 people with BSs in chemistry or biology and a garage and a couple $100 can start doing. Now, if they're doing mammals, that's tricky. They need a veterinarian. They need to know something about IVF in the mammals. If they're doing frogs, changing frog eggs, probably not so hard. If they're doing mosquitoes, getting a hold of mosquito pupae may be difficult, I don't know, but not all that hard. If they're doing bacteria, so for example, E. coli, everybody in the world has access to samples of E. coli. Every toilet contains lots of E. coli. You take that E. coli, we know the sequence, you use CRISPR to change that sequence to the known sequence of some of the pathogenic strains of E. coli. I don't know. I don't think it takes very much education, money, time, or skill for somebody to do that and release it. Now that's sort of the bioterrorism scenario. Bioterrorism, happily, has lots of other barriers, the delivery issues, the blowback issues, coming back in your own population, et cetera. This makes the creation of the pathogen, getting the pathogen easier. But what I worry about more is some of the inadvertent ones. People think they're doing something that will be good, and they release it. And it turns out to be kudzu, or some other nasty little pathogenic invader. My favorite example of this, how many of you know about the bird, the starling? Little black bird. Hundreds of millions of starlings in North America now. They fly in great big flocks, they're noisy, they're dirty, they're kind of a pest. They're not native to North America. In the 1880s, a Shakespeare enthusiast in New York City thought it would be nice if every bird mentioned in Shakespeare was in North America. So he released some breeding pairs of starlings into Central Park. And now they're all over North America. Somebody makes a change they think is going to be good, they release it. Maybe it's good, maybe it's bad. If there's a gene drive involved, maybe it spreads very quickly. Are we going to even know that's happened? Is our current regulatory scheme good enough? No. It's not good enough in two respects. First, it doesn't even regulate some uses of CRISPR. USDA recently said, not using CRISPR to knock out a gene that browns mushrooms after they're cut doesn't fall in its jurisdiction, because you're not adding a gene from another species. The mushroom doesn't have the CRISPR still in it. Gene drive would be a little bit different from that. So there's nothing changed. There's no regulation. Now, I think there probably doesn't need to be much regulation of a mushroom that doesn't brown, but I would like somebody to take a look at it, especially if it's about to be released into the wild. Right now, nobody does. But even more importantly, those regulations affect the entities that pay attention to them, that know about them, that can be reached. They affect Stanford and Berkeley. They affect Monsanto. They affect the Salk. Do they affect two guys in a garage? How do you even know what the two guys in the garage are doing, that they're using any of this? So I think the possibilities here for out-of-control changes of the biosphere are not likely to lead to Armageddon, but it would be nice if we had some knowledge of what was going on, some protection. We had people follow protocols in order to try to ensure against inadvertent release. Before there was a release into the wild, that there was some study and testing. National Academies just had a nice report on gene drive calling for this kind of testing in semi-wild before you release anything. But it's not law, it's just a National Academy's recommendation. And there is, to the extent the law requires that is really unclear. And also things for cleanup. If it goes wrong, have a plan, either a kill switch in the organism, or a plan for getting rid of them, or insurance, or something else. And none of that exists right now. People will do this for a wide variety of reasons, and many of them will be great. I think the changes to crops and livestock that can be done this way will be great for the environment, sometimes for the animals themselves. The polled cattle, I think, being a really nice example of that. They'll be great for, say, adapting to climate change. Getting more drought-tolerant, salt-tolerant crops, for example. This lets us do that in a much better, faster way. Those are the uses in non-humans people think about, but there are also the interesting, unusual ones, like changing mosquitoes to avoid human disease, either to kill them off or to make them resistant to the malarial plasmodium. Mosquitoes don't get infected with malarial plasmodia. People don't get infected with malarial plasmodia. And you've managed to cure malaria, at least until some other mosquitoes mutate around and pick it up, or the malarial plasmodium mutates around your change. I don't think there are any final victories in biology. Biology is too tricky for that. Those are uses people don't think much about. De-extinction, one of my personal favorites. Woolly mammoths, probably not so soon, in part because elephants are hard to come by and a 22-month gestation period is kind of a barrier. But passenger pigeons or other small mammals, I think we'll see de-extinction pretty soon. We're already kind of seeing it in the American chestnut tree, which is almost extinct thanks to a foreign blight, a foreign fungus. And people have genetically modified the chestnut to be resistant to this fungus. We'll see more things like that. We'll see some that don't seem so troublesome, like chestnuts, and some like saber-toothed cats or mammoths that will make people a little leery. We will not see Jurassic Park, because you need the DNA sequence, and nothing's going to survive 66.4 million years. But Pleistocene Park, I think you guys will live to see. I think I may live to see Pleistocene Park. And boy, would I pay a couple $100 to see real mammoths and mastodons and saber-toothed cats and giant ground sloths. I'd do it. So you'll see that. How many of you have or have ever had a dog? So any of you have the little tiny bread box dogs, Shih Tzu or Chihuahuas? Okay, 10,000 years ago, those were wolves. And we've degraded that species to that level. In 10,000 years, the old-fashioned way. Or we've elevated it, depending on your perspective. we'll make pets with CRISPR. We already have the glowfish, which is now legal in California after bizarrely being illegal for a decade. You know, there are fish that have red, green, blue, and other sorts of fluorescent proteins in them. We will make, now cats are tricky, because I don't know that cats are actually, I think cats domesticate us rather than the other way around. But we'll certainly continue to mess with dogs, with horses, with other companion animals. Gardeners. Anybody here a gardener? Anybody like roses? Anybody seen a blue rose? Only if they're dyed. No one has ever been able to breed a blue rose. I think CRISPR should be able to make blue roses. Shouldn't take very long. People will use it for that. De-extinction of foods. Those luscious old tomatoes and peaches, some of us remember from our youth, which probably weren't actually all that good, but may have gone extinct, could be brought back. So, and bioterrorism, just to go on the dark side. There are all sorts of uses of this in non-humans. Some would be good, some would be bad. We need to pay attention to them. The constraints against them are so much lower than they are against human uses that it really frustrates me that all the oxygen in the discussion is taken up by human germline. And 90% of the near-term risks, I think, are non-human. Okay, well, I think that's all I know about that I want to, oh, one last thing. It is true. So our society in particular lives on hype, and everything is hyped, whether we like it or not. And I'm looking at scientists, respectable, respected, appropriately respected scientists who may not hype their own stuff, but the Berkeley or Utah press offices, Stanford press office announces a breakthrough at least once a week. And if 1 in 100 of them ever turns out to make a difference in the world, I think we're doing pretty well.
Probe never.
Right, right. Once a minute. Are Eric's lips moving? So we live on hype, and some of the stuff is overhyped. There are enthusiasts, there are early adopters, there are people who are out there. There was a nice piece today in Technology Review, which is MIT's online magazine, by Antonio Regalado, who's a really interesting writer. Some of you may have contributed money to the Kickstarter to make glow-in-the-dark plants. They've now gone through $900,000 and they don't have a plant that glows. It turned out they picked a really hard target. They picked a bad target. That doesn't work. I predict some Silicon Valley billionaire will make a unicorn for his daughter within 20 years, his 12-year-old daughter. Maybe that won't happen, although I've been told recently that $5 million, $10 million, 5 years, probably can get one, which a Silicon Valley billionaire could certainly afford. Some of this stuff won't happen. Some of it will. Some things that we haven't talked about or haven't even thought about will happen. The non-human space is the space where it's more likely to happen because it's less regulated, less constrained, and that's where we're not paying attention and where we should. So that's all I know. Thanks so much for your time and attention.